Inhale. Exhale. We do this at least 22,000 times a day. That is why we want you to live your best lung life.
The Lung Life webinar series empowers us to live our lives and breathe easier.
Lung Saskatchewan has partnered with GSK to offer a free 5-part webinar series featuring key opinion leaders in their respective fields of expertise, all with one goal: for you to live your best lung life.
We know that education, treatment, and support are vital for lung health, whether it's to manage an existing lung disease or to prevent lung disease altogether.
I have learned something new on every webinar that I've attended with Lung Sask.
The Lung Life webinar series offers a wide variety of topics, including disease-specific presentations, as well as education on lung disease prevention.
The Lung Life webinar topics include sleep apnea, asthma and allergies, pneumonia, COVID, and influenza vaccines, quitting smoking, and everything you need to know before you go for a spirometry breathing test.
The knowledge we gain from science and research makes for a better and healthier tomorrow. It is constantly evolving. The Lung Life webinar series helps you stay informed. Do you know that 1 in 5 Canadians have lung disease? Asthma is common but serious, and one of the leading reasons for emergency department visits in children. 30% of Canadians are at risk of developing sleep apnea, a disease where you stop breathing in your sleep. And of course, anyone with lungs can become infected by viruses that leave us breathless. These webinars are not only for people with lung disease, but for anyone with lungs.
Knowledge is power, and it can help us all breathe a little easier.
To find out more about the Lung Life webinars and to register, visit lungsask.ca. Missed a webinar? Don't worry. The recordings will be on our website for you to view at any time. Live your best lung life. Visit lungsask.ca today.
Good evening. Thank you for joining us for tonight's Lung Life webinar, Pneumonia, COVID-19, and Influenza Vaccines 101. Prevention is good medicine, with scientist Dr. Alyson Kelvin. My name is Jill Hubick. I'm a registered nurse and certified respiratory educator with Lung Saskatchewan. It is my pleasure to welcome you and to be your host and moderator. Before we begin the presentation tonight, I would like to acknowledge the land we are hosting this webinar on is on Treaty 6 territory, the ancestral and traditional territory of the Cree, Dene, Blackfoot, Saulteaux, and Nakota Sioux, as well as the territory of the Métis. Lung Saskatchewan affirms our commitment to reconciliation and the Truth and Reconciliation Commission of Canada's calls to action. We acknowledge the many First Nations, Métis, and Inuit whose footsteps have marked these lands for generations, and we are so very grateful to be able to host this webinar where we are. Supporting and helping people living with and caring for people with lung disease is a priority for Lung Saskatchewan. We offer a variety of online support groups for patients and caregivers. The support groups include asthma, COPD, pulmonary fibrosis, COVID-19, lung cancer, lung transplant, sleep apnea, and Saskatchewan Quits to help people quit smoking. These support groups are here to help those living with lung disease or for those who want to quit smoking and navigate their journey in a safe, supportive way. Providing education and support is one way we hope to help you live your best lung life. If you are not a member of our Lung Saskatchewan support groups and you'd like to be, please visit our website at lungsask.ca. Following Dr. Kelvin's presentation tonight, we will have time for a moderated question and answer period. So if you have questions for Dr. Kelvin, please type your questions into the question feature on the webinar software. You'll find this feature in the control panel on the right-hand side of the screen. And if you want to watch this webinar again Or if you want to view any previous Lung Life webinar presentations, we hope you'll do so by visiting our website, lungsas.ca, where you can view the recordings. All right, it is now my pleasure to introduce our presenter. Dr. Alyson Kelvin is an early career virologist and vaccinologist at the Vaccine and Infectious Disease Organization. Her research focuses on emerging respiratory viruses of pandemic potential to develop broadly protective vaccines and to understand disease mechanisms such as long COVID. In 2003, Dr. Kelvin was part of a team investigating SARS-CoV infection in patients in Toronto. She has since studied many high-profile viruses, including H7N9, pandemic H1N1, historical influenza viruses, Zika virus, and SARS-CoV-2. Dr. Kelvin responded immediately after the identification of SARS-CoV-2, initiating studies in January of 2020 for COVID-19 vaccination development in Canada. This work is documented in the Nature of Things episode, The Great Vaccine Race. She is also a member of the World Health Organization's working group, COVID-19 Vaccine Development, and COVID-19 Assays, and has spoken frequently with the Canadian Parliament and Senate regarding COVID-19. We are so lucky to have her here tonight as a presenter. Dr. Kelvin's presentation is part of our 2023 Lung Life webinar series, which is sponsored by GSK. This educational webinar series was created to empower those living with and caring for those with lung disease to live better. This is the third year of our Lung Life series partnership together with Lung Saskatchewan and GSK. And together, Lung Saskatchewan and GSK will be bringing presentations on different lung health topics throughout the year. So I'm very pleased to welcome Marie-Claude from GSK to bring greetings tonight ahead of our presentation.
Bonsoir everyone, my name is Marie-Claude Desaulniers and I am the Patient Engagement Manager at GSK in the Patient Affairs and Advocacy team. In the scope of our work, we engage with patient organizations by listening to patients throughout the life cycle of their medicine, integrating those insights into our business planning to improve patient outcomes. GSK is very proud to collaborate with LungSask and to support the 2023 Long Life webinar series. LungSask is providing you with accessible, practical, evidence-based information and strategies coming from key leaders in their field of expertise. The topics of these webinars have also been chosen based on your feedback, which is so important to listen to in order to bring the relevant informations which we hope will help you live a better long life. Thank you very much and have a great evening.
Hi, my name is Alyson Kelvin, and I'm a scientist at VIDO, the Vaccine and Infectious Disease Organization at the University of Saskatchewan. I'm really happy to talk to you today about pneumonia, COVID-19, and influenza vaccines, really thinking about how prevention is good medicine. So a little bit about myself. I am an emerging virus researcher or scientist, again, at the Vaccine and Infectious Disease Organization. And I focus mostly on viruses that are newly occurring around the world and cause respiratory infections. I'm really interested in why some people develop more severe disease from these respiratory viruses than others. And I think this might have to do with age, gender or sex, as well as that person's genetics. The ultimate goal of my work is to focus on why these emerging respiratory viruses cause disease and how I can prevent this severe disease by developing better vaccines. So now I want to talk a bit about breathing and the respiratory tract before we get into viruses that affect the respiratory tract and the vaccines that we develop to prevent these viruses from infecting. So breathing and oxygen. Oxygen is an element needed by most organisms to grow, reproduce, and turn food into energy. Humans, we get our oxygen through breathing. We bring air with oxygen in it into our nose and mouth and then into our lungs. And here is where oxygen is exchanged for carbon dioxide within our lungs, which is ultimately delivered to our cells, where our cells use this oxygen to break down the food that it needs to live and grow. So looking more at our respiratory tract, we have an upper and lower respiratory tract. Our upper respiratory tract is our nose, our pharynx, our larynx, our nasal turbinates, and sometimes can be considered our trachea or windpipe. Our lower respiratory tract is our lower reaches of our windpipe or trachea, and then our lungs. Our lungs are delicate and need to be protected. We have 5 lobes of these lungs in humans. On the right side, we have 3 lobes, an upper, middle, and lower. And on the left side, we have 2 lobes, an upper and lower. So sometimes we only think of lungs as being 2-lobed, but it's actually 2 large organs that are divided into lobes. So for breathing, we take in oxygen into our upper respiratory tract, our nose or our mouth. It travels down our windpipe into our lungs, and where it can then be exchanged for carbon dioxide. We can only last about 4 minutes without breathing. That's how important oxygen is to us. And we take about 22,000 breaths a day. And we are really just powered by breathing, and we often don't think about breathing, and it's just controlled by our brain. So now thinking about in more detail breathing and gas exchange, I pointed out the pieces of the upper and lower respiratory tract. Here this diagram is showing the upper respiratory tract with the nose, nasal turbinates leading to the pharynx and the larynx, as well as the mouth being involved. These tubes then connect to our windpipe or trachea. Our trachea then moves down and branches into 2 bronchus, or bronchi plural. These are 2 tubes that reach into our lungs, and then they begin to branch, making even smaller branches, which we call bronchioles. And at the end of the bronchioles, this is almost like a tree, at the end of the bronchioles are these sacs that we call alveoli, and these are our air sacs, where the air goes that we breathe in with the oxygen. This is where the oxygen is exchanged for carbon dioxide. Importantly, lining these air sacs or alveoli are blood vessels, and that's where our red blood cells, which carry our oxygen or carbon dioxide, exchanges these 2 elements. And here this process is shown on a cellular level. What's important to know is that we again just have this blood vessel that lines the air sac or alveoli. Here I'm pointing to it with my laser pointer. And carbon dioxide moves out of our red blood cells, allowing oxygen that's inside the air sac to move into the red blood cells and leave, and then be delivered to our organs throughout the body. Something that I find really interesting is that although babies are born with lungs ready to breathe, they're not fully mature. They still have growth and development left to do. At birth, there's only about 60 million air sacs or alveoli in the baby's lungs, whereas in adulthood, we have about 300 million air sacs. So these alveoli mature. Most of the maturation is done by the time we are 4 years old, but maturation can still occur until we're in our 20s. This maturation and expansion of blood vessels, nerves, and areas of the alveoli allow for more gas exchange. So what's in the air that we're breathing? We know that there's oxygen in the air, but this is only a little over 20%. The rest of the air is made up of nitrogen as well as other elements such as argon and carbon. But within these elements that are in the air, there's also pathogens such as viruses. So these pathogens, such as viruses, are exploiting our process of breathing to get into our lungs, infect us, replicate within our lungs, and then move back out through our process of breathing, where the viruses can then infect other people. This is shown in this diagram where we have an infected person. They have virus infected within their upper and lower respiratory tract. As they're breathing out or coughing, they're releasing various virus particles into the air. The virus particles can then hang in the air until someone who is naive or uninfected comes along, takes a good deep breath, and then is exposed throughout their upper and lower respiratory tract to these viruses, and they might be infected. So now I want to talk a bit about pneumonia. It's a serious disease that does affect the way that we breathe.
Pneumonia is inflammation in the air sacs of the lungs, or your alveoli. It's caused by infection of various different types of pathogens, such as viruses, bacteria, or fungi. Because pneumonia is caused by an infection, it can be contagious, meaning one person can give it to another person. It can affect both sides of your lungs or even a single lobe on each side. It can affect any individual, but there are people who are at higher risk, such as the vulnerable populations of older individuals, younger individuals, those who are immunocompromised, people with chronic health problems such as asthma, diabetes, or heart failure.
So the symptoms of pneumonia are quite similar to other infections or what we call influenza-like illness, but they will be specific to our lungs and our respiratory tract, as well as we might have more systemic symptoms such as headache. So we might have headaches, fever, chest pain due to the infection within our lungs, have coughing up sputum, and might show our illness through pale skin. How do we diagnose pneumonia and treat it? Well, pneumonia must be diagnosed by a doctor by running multiple tests. One of the tests might be an X-ray, as shown here. Typically, we think of healthy lungs and an X-ray to be kind of dark black spots, and we know that pneumonia is there when we see lighter patches that are often called, a technical term, ground glass opacities. So what's causing that more lighter texture and tone that we're seeing in this X-ray? Well, what happens when we have pneumonia, we have our alveoli sacs, but with the infection, unfortunately, these sacs start to fill up with fluid, thereby causing it hard to breathe, hard to have that gas exchange, and further, the fluid is what's showing up on these X-rays. So now it's September, we're getting into the cold and flu season. So brace yourself, we're here, we're going to be exposed to different respiratory viruses. So when is the cold and flu season? Cold and flu season occurs from about September to March, sometimes lagging into April, in our northern hemisphere. It's important to recognize that this is over our fall and winter, and cold and flu season might be different depending on where you are in the world. So what are the symptoms that we would often see during cold and flu season? Again, we might call this influenza-like illness, but this is a range of symptoms that you might associate with a cold or flu and a respiratory tract infection, such as fever, cough, sore throat, runny nose or stuffy nose, muscle and body aches, headaches, fatigue, and sometimes gastrointestinal issues such as vomiting or diarrhea. But is there a difference if we look a little closer between having a cold and having the flu? Well, actually, colds are typically considered mild and very common, whereas the flu, being also highly contagious, it's a bit more severe. And if we look at the specific symptoms and how they and their onset, we see that colds, their symptoms come on quite gradual. It's rare to have a fever, and again, we think of colds as being a bit more mild. Aches and pains are also uncommon, or you'll only have a little bit, along with chills and weakness. As well, it's not as often that a cold will cause a lower respiratory symptoms in your lungs and chest. When we compare this to the flu symptoms hit you almost like a truck. It's abrupt. All of a sudden, you feel like you're down for the count. You might have a sudden fever, a sudden cough, runny nose, or stuffy nose. As well, even though you might have some nasal symptoms, they won't be as strong as what you would have with a cold. So now these are general differences between a cold and flu that are important to pay attention to if you have a respiratory tract infection. And importantly, both colds and flus are caused by respiratory viruses. So I've talked a bit about viruses so far, but what is a virus? Technically, if we get down to the technical definition, viruses are submicroscopic obligate intracellular parasites. What does that mean? Well, if we think of submicroscopic, we think we might need a microscope to see something that is microscopic and we can't see at the naked eye. Well, a virus is so small that you can't even see it with a normal microscope. So that's why it's called submicroscopic. You have to use an electron microscope to actually see it. An obligate intracellular parasite, let's break that part down. Obligate means that it needs to be intracellular, inside the cell. And in that, because it needs a cell to have activity and it needs to be inside that cell, we think of virus as a parasite. So taking that definition, here's an electron microscopic picture of a cell that's been infected with virus. And you can see I'm outlining the outside of the cell with the laser pointer, and inside are all these small dark dots, which are viruses. So viruses, again, are inside our cells until they break free, where they're then trying to infect more cells. So looking at that in a cartoon, this is a life cycle of a virus. Here we have a circular virus that is entering in a cell, and you can see the cell now. I'm outlining that with my laser pointer. It gets inside the cell. At this point, the virus is active. It can make more of itself. And as it's making more virus, it wants to get out and escape to find new cells or new people or animals to infect. And by understanding how a virus gets into a cell, we often use that as our base to develop vaccines. So what are the viruses that cause colds and flus in our cold and flu season? Well, it's almost obvious for the flu or influenza. Influenza viruses cause the flu. Influenza viruses are not one virus. Interestingly, it's a huge family, complicated family of different viruses. They're all related, that's why they're all influenza viruses, but there's many of them. And they're broken down into 4 different types. And I've shown that here in the diagram. We have type A, B, C, and D. And fortunately for us, we only have to worry about 2 of those, type A and B. They infect people and circulate in human populations in the cold and flu season. On the reverse, looking at what causes the common cold, there's a large number of different viruses and virus families that cause the common cold. Some of the more familiar ones will be respiratory syncytial virus or RSV, rhinoviruses, coronaviruses, which we've heard a lot about lately, adenoviruses, parainfluenza viruses, and metapneumoviruses. And interestingly, even though they all cause respiratory tract infections during our cold and flu season, they don't all cause cases to peak all at the same time. People can only really be infected with one virus at one time. So they kind of take turns and they have different waves throughout our cold and flu season. And I'm showing you that with graphs from the Respiratory Watch, which is put out by the Public Health Agency of Canada, showing our peaks and valleys of cases caused by different respiratory viruses through the cold and flu season last year. So here we have SARS-CoV-2, influenza A, influenza B viruses, RSV, rhinoviruses, and you can see, some of these viruses like influenza A, they really peak right in our last week of the year or our first week of the year, which is December and January, and that's where we think of when we usually get hit hard with the flu. Conversely, rhinoviruses, which cause the common cold, are peaking a little bit more in the September months, September and fall months, as well as in the spring months. So you can see rhinoviruses are taking turns with influenza viruses. And if you look through the rest of the graphs, you can see each type of virus has its own pattern of infection. So now we need vaccines to prevent disease that are caused by infection with these viruses. So what is a vaccine? Let's talk about that first. Well, a vaccine is made or developed with the purpose to prevent infection. It's to teach your immune system or immune cells what a virus looks like before you even come into contact with it. So I like to think of that almost as a general in the army training its army about the enemy before they even go to battle and have met the enemy. So when you get a vaccine, you're teaching these immune cells what the virus looks like, and the specific cells that you're teaching are B cells. B cells are important because they make antibodies. Their antibodies will be specific to that virus. And T cells. T cells will look for cells that are infected with a virus so that they can clear those infected cells so you only have healthy cells inside you. Importantly, the antibodies that are made against the virus by B cells after you've been vaccinated can surround a virus. So here we have a virus surrounded by antibodies binding to it. And these antibodies that bind to the virus, they block the virus from getting into your cells and infecting them. We call that neutralization. So now let's look a little bit deeper into influenza. So influenza is really a global health problem. It affects all parts of the world. We have about 40 million cases worldwide per year, and this is associated with between 500,000 and 650,000 deaths per year. Strikingly, as we often think that it's just influenza, it's a regular disease that we get. Actually, over 3,000 people die each year during our influenza season from an influenza virus infection. That makes my heart sad. So let's look a little bit more at what type of disease influenza viruses cause. Again, it's a respiratory disease, and interestingly, it can range in severity from mild to severe depending on the person. In severe disease, it can cause pneumonia, respiratory complications, which might require ventilation within a hospital, and this can even lead to death. Our typical symptoms, as we went over before, can be headache, runny nose or stuffy nose, coughing, body aches, and fever. And vulnerable populations are more susceptible to developing those severe outcomes, such as pneumonia and other respiratory complications. So influenza, what does that mean? The name influenza came from 15th century Italy, where there was an epidemic and the people didn't know what was causing this epidemic. And they thought, because, it came on seasonally, this must be the influence of the stars. So influence became influenza, and that's how influenza got its name. Influenza has also gone by some other historically humorous names, including a jolly rant, a new delight, a fashionable illness, and unfortunately, during the 1918 pandemic, which led to between 50 and 100 million deaths, influenza was also called the Blue Death. So what actually causes influenza? We've talked about it being a virus. We've talked about it being a large family. We're looking a little bit closer at influenza A and B viruses. Influenza A viruses are broken up or named by their surface molecules. Here in this diagram, we can see a cartoon of an influenza virus. On the outside, we have these large, almost spikes that we call hemagglutinin, or an H. And these H's are numbered 1 to 18. As well, we have these smaller spikes called N's, or neuraminidase, and they're numbered 1 to 11. And for influenza A, that's how we get their names, by what combination of H's and N's this virus has. So I'm sure we're all familiar with H1N1 and H3N2 at this point. These are the two, what we call subtypes or combinations of H's and N's that infect people. Influenza B can also infect people, but they're named by lineages, B. Yamagata and B. Victoria. What's important here that I have to show you is that these are the epidemics or influenza seasons that have occurred in Canada through a 10-year time span. And each peak shows the number of influenza cases, and it's broken down by what kind of influenza are those cases, where influenza B cases are shown in this brown, and influenza A cases shown in greeny blue. And as you can see, that each year there's a different proportion of what cases are broken down into, if these are H1N1 cases or H3N2 cases, which means that the virus composition in our environment and what's being transmitted from person to person changes each season. And also, which isn't as obvious from this graph, the virus often mutates and becomes a little bit different each season. So from season to season, you'll have mutations in these H1N1 viruses and these H3N2 viruses. So it's constantly re-emerging and emerging, and so people are continuously exposed. So the influenza viruses mutate. How does this happen? Well, here I have another cartoon of an influenza virus with its H's shown in red, and the blue represents the N's. And then inside we have the genome or genetic pieces of the virus, and that's where the mutation occurs. Importantly, as the virus is making more and more of itself when it infects people, it can make mistakes. And these mistakes might lead to a change in one of the genes. And here I've represented a single change, and this might lead to a slight change in its H protein on its surface. So here the H is in red, but after this mutation happens, the H might turn to green. Interestingly, because influenza viruses have its genetic material in these segments or pieces, it can actually swap gene pieces with other influenza viruses. And this is what we call an antigenic shift, whereas a single point mutation we call an antigenic drift. So once, if by any chance these viruses infect the same cell at the same time, they can swap different pieces. So here we have a virus that has black gene segments and another influenza virus that has blue gene segments. And after they're done infecting and then they get put back together, we can have a total mixture, here black and blue gene segments in a brand new virus. Often, and this process is called antigenic shift, often this process might lead to a pandemic because this is a brand new virus that's been created, which means it can rapidly infect people who have never seen this type of virus and transmit quite quickly across the world. So this means we need vaccines to prevent influenza viruses from infecting us, leading to pneumonia, as well as other respiratory complications. So what's in our seasonal influenza vaccines? As we discussed, there are two types of influenza viruses that infect people annually. This is influenza A and influenza B. Within both of these are two subtypes or lineages of influenza A and B viruses that circulate within people. So for the A viruses, we have H1N1 viruses infecting people seasonally, as well as H3N2 viruses. And for influenza B, both of their lineages can infect us seasonally. So we have B Yamagata lineage and B Victoria lineage. Now, because these four kinds of viruses are circulating in people every year, we need to have a piece or a component representing each one of these types of viruses in our vaccine. So annually, our most effective vaccines have representative of the four different kinds of influenza viruses. And importantly, vaccination has been shown to decrease infection. So if you're vaccinated, you're less likely to be infected. You have decreased severe clinical symptoms and decreased hospitalization. If you do get infected and you've been previously vaccinated, you have a decreased amount of virus in your lungs, so less chance of developing pneumonia. As well, you have less transmission, so you're releasing less amounts of virus if you cough or sneeze, which means that there's a less likelihood that other people who aren't infected could be infected by you. So it's really important in that, as we talked, that influenza affects people seasonally in these epidemics, cold and flu season, as well that the virus mutates quite quickly and changes year to year. This means that we need to update our vaccine almost yearly. So how does this happen? We have surveillance centers all over the world that's coordinated by the World Health Organization. There are 144 of these national influenza centers in 144 countries throughout the world. They collect samples, they sequence the viruses, they look at how the viruses might infect cells, and then the data is reviewed from these 144 centers twice yearly, once in the late winter, spring, and once in the fall. And when the data is reviewed, what we're trying to do, or what these surveillance centers are trying to do, is determine what viruses should go in the next year's vaccine. So they try to decide which flu viruses are making people sick, what virus is most represented, what is causing the most influenza cases, was last year's vaccine effective, and most importantly, was if we used last year's vaccine on this year's most prominent circulating viruses, will these be protective? So after these gatherings, answers to those questions are determined, and then it's decided if the vaccine should be updated in one of the components, either the H1N1, the H3N2, or one of the Bs. So what do we have this year? Well, these are long names, but I just thought that I would include these in my presentation just for those curious minds out there. If you wanted to know what strains are in our 2023-2024 influenza vaccine that we're using here in Canada. For the H1N1 component shown here, where it's represented by a virus called A Victoria 4897 from 2022. Our H3N2 component is called A Darwin 9 from 2021. And our B Victoria component is A Austria 1359417 from 2021. And B Yamagata is A Phuket 3073 from 2013. And so we break down these, what we call strain designations, by if it's an A or a B virus, where was this virus detected? So this is a place. What number of our sample was it that this virus was? So for this example, it was the 4,897th sample collected from Victoria. And the last number is the year that it was collected, so from 2022. What's important here is to just see that in the year, we have had to more recently update the H1N1 component because it's from 2022, whereas H3N2 seems to be more stable right now, not being updated since 2021. And if we jump down here to B Yamagata, it hasn't had to be updated since its 2013 strain. So how do we forecast what's coming up for the influenza season? We're just starting ours, and people always want to know, how severe will it be? What will our Canadian 2023-2024 influenza season look like? Will we have a lot of hospitalizations? Will the vaccine be effective? So often we look to Australia, and as I mentioned earlier in the talk, our northern hemisphere sees our cold and flu season during our winter months, which are between September, October to March, April, whereas when we look at the southern hemisphere, such as the country of Australia, they see the flip side. They have their winter months during our summer months. So they are just right now coming out of their winter. So we can look and see what their flu season looked like. So here I have a graph of influenza cases from Australia over a number of different recent years. The most recent one, our 2023 season, the number of influenza cases are represented by the red line. And as you can see, there's less influenza cases in Australia this past year than what they've seen in the past. But it was mostly, if you break down what type of virus was causing these infections, it was thought of to be an H3N2 season, meaning H3N2 viruses were causing the most influenza cases. As well, infection was highest in kids between the years of 5 and 9, and the majority of infections were caused by viruses matching the vaccine. So this is actually really great news for us. It means that if we get our recommended, WHO recommended vaccine, we'll most likely be protected by the virus that's circulating because the virus matches the vaccine that was recommended. But it's important, even though we see less cases than what was normally seen, we can't always predict influenza. We might have a completely different season, but also more importantly, this virus seemed to affect kids between 5 and 9, and we do want to protect them. The best way to protect kids, as well as everyone else, is to get vaccinated. And those who can't be vaccinated, if we can vaccinate and decrease the amount of infections and the amount of virus that's in our communities, we are then secondarily protecting those people. So now let's look at another major respiratory virus that's caused quite a bit of problems in the last couple of years. So this is the family of coronaviruses. And what's interesting is that coronaviruses can cause the common cold, but it also can cause very severe disease, including COVID-19. So here, as similarly as what we saw with influenza viruses, it's a large family made of 4 different types again: alpha type, beta type, gamma, and then delta. We know that our common colds can be alpha as well as beta coronaviruses, and I've listed them here. You don't necessarily need to know these names because we don't talk about them often, but if you're curious, these coronaviruses that frequently cause colds in people are called 229E, NL63, OC43, and HKU1. Now, even though these were mostly mild, coronaviruses can also include other viruses that can cause very severe diseases. So the first one that we saw was severe acute respiratory syndrome coronavirus from 2003, so SARS-1. That was the first time we found out that not all coronaviruses are just mild colds. And then again in 2012, there was an outbreak of MERS coronavirus, Middle East respiratory syndrome coronavirus. And then, of course, we saw our third outbreak of a very severe coronavirus that led to a pandemic that we've now called SARS-CoV-2. And this graph shows the waves, seasonal waves of coronaviruses causing common colds. So here we have in March, April to May, our common cold cases are tailoring off. But as we go back into our fall and then winter, they rise again. So this is what our cold cases looked like caused by coronaviruses before we had COVID-19. So this is for the year of 2019. Importantly, what comes out of this is we can see that there was seasonal epidemics and that there was no vaccine, no coronavirus vaccine in this time to prevent seasonal colds. And even though most cold virus infections in most people might have been mild, in some people, such as the vulnerable and older individuals, an infection with a common cold coronavirus could lead to a lower respiratory tract infection, even though in healthy adults we saw it mostly in the nose and upper respiratory tract. And since there's infection in the lower respiratory tract or the lungs, this could lead to pneumonia. And now if we look at SARS-CoV-2, so SARS-CoV-2 emerged in 2019, and scientists think that this virus came from a bat, possibly started to infect a secondary host, and then led to spillover in people. And of course, once this virus infected people, it then led to rapid transmission, cases that were severe, leading to hospitalization, and then of course a global pandemic. So why are coronaviruses called coronaviruses? Well, here's our schematic of SARS-CoV-2. We have spike proteins along the outside, and these spike proteins led to the name. When scientists saw these spikes, it led to the name coronaviruses because scientists thought it looked like a crown of spikes. Again, SARS-CoV-2 is most related to SARS-CoV-1 from 2002-2003. And so far, SARS-CoV-2, which its infection causes coronavirus from 2019 or COVID-19, has led to an estimated 20 million deaths worldwide. And after infection, if you are able to recover, long COVID is now becoming a serious concern. So again, looking a little bit more at the diagram, we see the outer spike protein. This is where the virus attaches to our cells, and inside we have a genome or genetic material. There's also other proteins that are important to the virus, but the one that's most immunogenic and most interesting for vaccine design is the spike protein. So again, with COVID-19 and infection with SARS-CoV-2, it can lead to a range of disease severities from mild or asymptomatic. And even though these people might have evidence of lower respiratory tract involvement, such as pneumonia, after having an X-ray or CT, their symptoms might only be mild, but it can also be severe, lead to severe pneumonia, as well as something that we call ARDS, or acute respiratory distress syndrome, sepsis, multi-organ failure, and possibly even death. And again, there's a list of higher risk groups. So now let's take a little bit deeper look into COVID-19 vaccines. So as I mentioned, this is another schematic of SARS-CoV-2, the virus, where we have these outer spike proteins or the coronas of the virus. This is where the virus this is the protein the virus uses to attach to our cells, and they do that through a molecule which we call ACE2. And this attachment allows the virus to get into a cell and infect it. So that's why we are very interested as scientists to develop vaccines against this molecule. So once a person is vaccinated, after learning about what a spike protein looks like, their B cells can develop antibodies that specifically bind to this spike protein, when it binds to this spike protein, we can have neutralization, as I mentioned earlier, and also this binding of this antibody physically blocks the spike protein from binding to the ACE2 receptor. So when SARS-CoV-2 first emerged, we knew a lot about coronaviruses because of our study in common cold coronaviruses, but also on SARS-1 as well as MERS coronavirus. So we knew the spike protein was a great target, but what platform should we make this vaccine again on? So when vaccines started to be developed, they were developed on 4 different types of vaccine platforms. They were developed on nucleic acid vaccine platforms, which include DNA and RNA, protein-based vaccine platforms, which might be protein subunit or virus-like particle, whole virus. This was kind of how we made vaccines. Our first generation vaccines, when we were first developing the field of vaccinology, where we would take an entire virus and either we'd weaken it so it would no longer cause disease, or we'd completely inactivate it. So then we didn't have to use sophisticated biotechnology to make pieces of the virus for a vaccine. And finally, vaccines were made on a viral vector platform. This was using a virus that was rendered so that it could no longer cause disease, and we'd put a piece of the spike protein within this viral vector, and the viral vector would almost be a delivery mechanism to our immune system to show our immune cells what that spike protein looked like. But of course, the most effective vaccine that we've seen so far has been developed on the nucleic acid RNA platform. So how does this work? We have pieces of RNA being delivered to our cells. Here's our cell. The RNA shown here is put inside a lipid nanoparticle. So this is kind of like a fat drop, and we put pieces of RNA in. The fat drop protects the RNA, delivers it to your cells so that the RNA can get inside. The fat droplet breaks up and only the RNA is left. The RNA is then used to make a piece of a spike protein, and then that's used, the spike protein is then used to train our immune cells, shown here, our T cells and B cells, on how it should attack SARS-CoV-2 virus if it sees it. The big benefits of using RNA vaccines is that they were easily scaled up, so we can make them fast, and we can make a lot of them to be shipped out across the world and hopefully stop or slow our pandemic. It was also able to both stimulate a T cell and B cell response, whereas some of those other vaccine platforms we talked about really only would stimulate a B cell response. So our 2 main vaccine companies that were making these RNA vaccines are Moderna and Pfizer, and both of them were targeting the spike protein in what we call a prefusion stabilized spike, or S, which means that the spike protein that was being made is no longer really able to break apart. It was quite stable if it was made inside of us. But we have a new problem, unfortunately. The first virus that emerged from China in 2020 or 2019, which we call the original SARS-CoV-2 virus, started to mutate. And yeah, we've seen this story before with influenza viruses. We now have different types and different flavors of this virus, and we're seeing them cause new waves of COVID-19 with SARS-CoV-2 infections. And these first main variants that we called them, or mutated original SARS-CoV-2 viruses, were Alpha, Beta, Delta, Gamma, and Omicron. So we have our vaccines at this point developed to the original SARS-CoV-2, but now we have the new problem of these new variants. What do we do? Well, we update our vaccines. So the first generation of COVID-19 were made against ancestral or original COVID-19 or SARS-CoV-2 spike protein, but now we have updated vaccines. So we have second generation vaccines that target the Omicron subvariants. Moderna has three different types of updated vaccines. One which we call bivalent, so it has two different representations of SARS-CoV-2 viruses in it. It has the original SARS-CoV-2, and then it has an Omicron BA.1. It has another bivalent, which has original and then BA.4 or 5 Omicron, and then it now has an updated monovalent Omicron vaccine, which only has the Omicron subvariant XBB.1.5. Pfizer also has updated vaccines which really mirror the first two from Moderna, and they do not have a monovalent Omicron XBB.1.5. So again, similarly as what we saw with influenza vaccines, COVID-19 vaccines have been shown to have similar benefits. So when you're vaccinated, you're less likely to be infected. If you are infected, you're less likely to have severe clinical symptoms, which might lead to hospitalization. You have a lower viral load within your respiratory tract, which means you have a lower amount of virus that you're transmitting to other people when you cough and sneeze. And really important, we've seen protection from long COVID. In people who are vaccinated, they're less likely to develop long-term symptoms of being infected with SARS-CoV-2, which we call PASC, or post-acute sequelae of COVID-19, also known as long COVID. And together, I would really like to thank everyone for listening. I'd also like to thank LungSask for inviting me to talk to you about this important subject. Have a good evening.
Thank you so much, Dr. Kelvin, for the wonderful presentation. And I also just have to thank you for the amazing work that you do each and every day. We're so lucky to have people like you that do what you do every day to keep us all safe. So thank you so much. I'll invite you to join us if you can. Turn your camera on and your microphone. There you are. And we will get right into the question and answer period. So I'll remind everyone that if you have questions for Dr. Kelvin, please go ahead and type your question into the software there. There's the question feature, and we'll do our best to get to all your questions. And again, a reminder that the presentation tonight was recorded. So if you missed something or you want to share this with friends, family, you want to watch it again, you can. It will be posted on our LungSask website at lungsask.ca. Okay, so first question, and this is actually a question just from me that I'm curious about. Dr. Kelvin, if you could just share, because I can honestly say until I met you, I've never met anyone with a career like yours. How did you get into doing this type of work and why?
Well, thanks for asking. I do also find it interesting to hear how people got to where they are in life, because we need so many different specializations. Me in particular, I've always been interested in viruses. Maybe it was when I think about it, I think about how when I was young in the 80s, there was a mysterious disease, that seemed to be only affecting gay men, which was then later turned out to be the HIV virus, which we know can affect anyone. And but it was a mystery and really important to solve this to save these people. So I was very interested in viruses after hearing that on the news when I was a young kid. And I think the influence of that, and my father, who's also a scientist, I almost grew up in a lab. So I was interested in being a scientist almost from when I was 2 years old and first learned how to talk. I'd tell my mom I wanted to be a scientist. So that's where the scientists started, and then I got interested in viruses during the HIV times. And then during 2003, when SARS-1 had an outbreak in Toronto, I was working as an undergraduate research student in the Toronto Hospital Network. And it was really interesting because we had this, again, new mysterious disease that was affecting the city. And there was a lot of fear in Toronto at the time, and especially because the virus was spreading throughout the hospital network, affecting a lot of healthcare workers and hospital workers. They asked, as summer students, they asked us to participate in some of the infection control. So they asked us to kind of handle the doors of the research institute, and we would screen people as they were coming in. And this is 2003 again. And so we'd give them hand sanitizer, we'd ask them if they had been out of the country, we'd ask them if they weren't feeling well, and then also take their temperature. So that's the first time I really saw an effect of how an emerging new virus could affect my world. And then I was lucky to be part of a team at the same time who was researching why this virus was making people sick and killing them. So we actually found people who did very unwell during their SARS-1 infection had an increase in a certain type of immune mediator called CXCL10, which affects your T cells. And that kind of is a good biomarker, and has really, once SARS-2 then emerged, I had all this previous experience and knowledge, and it was almost like reliving what I had gone through in 2003. And really after 2003, I was building my career to stop the next coronavirus and the SARS-2. So unfortunately, I feel I failed, but big global effort, I think, and hopefully we can prevent the next emerging virus.
Oh my goodness, your journey is just fascinating, and we're just so lucky to have you not only in Canada, but right here in Saskatchewan. Just thank you for all you do, and for, your curiosity. Who would have thought, from a young kid, and the influence you had from your dad, and then the experience you had as an undergrad, and what you're doing today. Wow. So thank you, and thank you for sharing that. I really appreciate that. Okay, so we've got quite a few questions here. One of the questions is asking, you talked about the importance of getting influenza vaccines as well as updated COVID vaccines when they're available. People are asking because they're usually available around the same time in the fall. Can people get their vaccines at the same time? Do they need to spread them out? If so, which one do you get first? Is it just whatever you can get available? What advice do you have there?
Yeah, so I think it's a really good question, and a question that almost anybody would have or should be thinking about. As a vaccinologist and an immunologist, my expertise says that you can get multiple vaccines at the same time and they're equally as effective. But really the regulation of how you get your vaccines and when is mostly done by the provincial health system. So some provincial health systems have recommended that children get space out their influenza and their COVID-19 vaccines, not because they're less effective if you get them together, but if a child is getting their COVID-19 vaccine for the first time, you don't want to be confused if they have any kind of complication with getting their first COVID-19 vaccine because they got it at the same time as their influenza. So, you know, some people can be allergic to vaccines. We want to know which one that person might be allergic to, even though that's a very, very rare event. You just want to understand your child the most. But immunologically, the majority of people would do very well with having both vaccines at the same time. And I got both of mine at the same time last fall, and we'll be doing it again this year.
Oh, wonderful. Thank you. That was wonderful, practical advice that I think we can all now take with us. So thank you. Okay, there's some questions you highlighted in your presentation, the mRNA vaccines. And first of all, I just want to thank you so much for explaining very complicated, topics, and you covered so much, but you did it in such a way using plain language that we could all understand. So thank you so much. But you talked about in the mRNA how they were very successful for COVID vaccines, in that they worked on both the two different types of cells, right, to be more effective, and then how they could make them quite quickly and make a lot of them, which is what we needed during the pandemic. And then you highlighted the Moderna vaccine and Pfizer vaccine. They're both mRNA vaccines. Other than they're made by different companies, is there much of a difference in them?
So the dosage is different, and as well as the time in between your vaccine doses if you're getting a primary series. So I guess scientifically, when you look at the vaccines and what has been done, you know, preclinically, the vaccines seem to be quite on par, and it doesn't matter, you know, even though the Pfizer vaccine, it has a much lower dose than the Moderna. If you lowered the Moderna dose, it would work fairly equally to the Pfizer vaccine. But the Moderna vaccine is safe in its higher dosage, which is how it is formulated right now. So if we get into the details of that, the Pfizer vaccine for adults is 30 micrograms, whereas the Moderna vaccine is 100 micrograms, so more than 3 times what you would be getting in a Pfizer vaccine. And then it goes down from there for children under 12 and then children under 5. So depending on which vaccine, you get approximately half of each dose as you go down.
Amazing. Thank you. I appreciate you. I mean, that's a lot of information that's always changing, and we're kind of tapping a little bit into the public health side too, and we understand you're very much involved at the scientific level, and so thank you for that and for sharing that with us. We really appreciate that. Okay, there's some questions about, can you explain how often you recommend right now getting a COVID vaccine, and with that, why do we need to get them every so often? Is it because the vaccine weans over time, or is it just because of the new variants? And what are your recommendations around that?
Mm-hmm. And I think that's another good question. And is a bit complicated because, as you know, we discussed, influenza is quite seasonal and regular, and right now SARS-CoV-2 isn't so regular and seasonal, and we don't know what variant might come along next. But what we did see with the mRNA vaccines is that they don't seem to keep your antibodies as high for as long as what we would have seen with a traditional protein-based vaccine. But saying that, you know, when you have different waves of SARS-CoV-2, you might only need that protection throughout the wave unless another wave comes along. So there's multiple factors going on. And what I would recommend right now, what I'm thinking is I'm hoping that SARS-CoV-2 as a virus settles down, we start to see that seasonality in it. So I would recommend getting your vaccine as we're getting into the fall and the cold and flu season, which, the spread of respiratory viruses happens more often because of many factors, large gatherings, people indoors more, the change in the humidity. They all help the virus from spreading more often and to more people. So I would say to protect yourself going into the cold and flu season, I would get my updated booster, which should be available after October 10th, I'm hoping, for the new COVID-19. And so that's what I'm looking forward to right now.
Oh, wonderful. Thank you. Yeah, I think many of us are looking forward to that. So thank you for that great explanation and great advice and practical advice that allows us to know how we can protect ourselves. So thank you so much.
I do think just if a new variant comes along, or if we have a new huge wave, I would also want to be ready for, if we need something in March, we might want to be thinking about a booster. But right now I'm trying to be optimistic that this turns out to be more like a seasonal update, like the flu vaccine that we can get going into the cold and flu season.
So with that, do you think that it's most likely moving forward that in the future that it will always be similar to influenza where we'll be getting yearly vaccines, or do you think there's been some chatter in the media of dreams in the future, hoping that we can have a vaccine that lasts much, much longer and is effective for many, many variants to come? What are your thoughts on that? I'm looking for your crystal ball there.
Well, I actually really, this is, the source and focus of all of my research is to develop a vaccine that's protective, that leads to protection over a longer period of time, both by boosting your immune system to have those antibodies for longer, but also antibodies that can recognize variants, so the virus as it's changing over time. So this is kind of, again, a multi-factor problem where we want to boost our immune system so that we have that protection for longer, but also that's broader protection. So that's what I'm hoping that we can solve at VITO, and possibly, maybe the larger companies might also come out with a vaccine formulation that does induce more broad and longer lasting protections. So I am hopeful for that.
Wow, that would be amazing. Well, I think we'll cheer you on for sure. And wouldn't that be neat in the future to do a webinar talking about this discovery? That would be just amazing. Okay, so you talk about that's part of your research right now, but I know that you've got your hands in many different projects. Some people are wanting to know if you can share, I hinted at this a little bit in your bio, about some of the work that you're also doing in long COVID.
Sure, I would be really happy to. So I think we had an absolutely fabulous and successful project on long COVID, which was really based on the wonderful contributions that we had from the community and our volunteers. So I have to say thank you to everyone who volunteered in our study. Me specifically, we had both a survey-based long COVID study, and from that came many other studies where my my real focus in that was trying to understand how the immune system was misregulated during long COVID. So focusing on that part of the project, we had about 200 volunteers, people coming into Vito, giving us their blood sample, as well as filling out a small questionnaire telling us about their COVID-19 experience, some of their health history, and then also how not only their experience with having acute COVID, you know, their severity while they were sick, but what type of recovery they had. Did they recover after 2 weeks? Did they recover after 4 weeks? Do they still have symptoms 2 years later? And what we found really interestingly is that people who felt like they had long COVID had lower amounts of virus neutralizing antibodies than to both the ancestral or Wuhan virus, as well as the Omicron BA.1 virus. So that was quite interesting. And that's compared to people who had COVID and felt that they recovered. So those people had much higher virus neutralizing antibodies. So you would have expected that if antibodies weren't somehow playing a role, then those antibody levels would be the same. But unfortunately, people who feel they have long COVID had lower amounts, meaning that their antibodies have a poor ability to encapsulate that virus and block it from infecting their cells. So that is concerning to me, or it gives us a hint maybe of what's happening, in that maybe they had a poor response to clear the virus, but also it's concerning because they want to be thinking about protecting themselves from another exposure of SARS-CoV-2 so that they don't get it again, because they might get it more easily because they have that lower level of antibodies. We also found a difference between females and males, where females seem to have, who felt they had long COVID, they had high levels of inflammation markers over time, whereas males seem to, after they had COVID, their inflammation went down, but unfortunately females seem to stay high even over 2 years. So I think that that's an important finding, because it might identify, and of course, you know, we're in early stages, but it might identify something that we could target. So those signals of inflammation, we could possibly target that, and that might help in recovery.
Wow, that's just fascinating. And yeah, thank you so much for sharing that and the work you've done in that area. And thank you to all the participants that took part in this study. That it can't always be easy to take part in those and be the patient, but it sounds like already, you know, you've learned something from there. You're already thinking about, you know, maybe how can we make a difference, make changes for the future. And I also really appreciated you highlighting in your presentation how getting a vaccine can also help prevent long COVID, which prevention, as you said in the very beginning, is some of the best medicine. So thank you. Okay, so I know we have tons of questions. I think we have time for one more. Okay, there's some people that really don't like getting needles. And so they get a little apprehensive getting a vaccine. And there's been some studies that have been done about influenza and other types of vaccines where it's given a little bit differently, like through the nose. Can you share if that's something you think that we'll maybe see in the future, and how that would even work?
So there is the flu mist, which is the influenza vaccine that you can get as a nasal spray. The data shows that it works best in children, which is great because a lot of children are afraid of needles. And, you know, I gave it to my kids when they were little, and they really appreciated it. But as we get older, because we've had too many experiences with influenza, it's not as effective as our intramuscular vaccine. But with COVID-19, there is a lot of research into trying to develop an effective either intranasal or intratracheal or a vaccine that will target more the lower respiratory tract. And I think that's really important because I think that that's your site of exposure to the virus, and that's where you want your immunity to be ready to go. So it's an easy way of establishing what we call memory immune cells to be sitting there ready to go if they see that that virus again, or something that looks like that virus, to protect very quickly. There is a lot of research going into that, but because it's the lungs and we know how delicate it is, that's also, you know, a concern. And I think that the advancements will be slower, but I'm still hopeful because we do see such promise with the influenza flu mist.
Oh, wonderful. Thank you. Yes, I'm sure a lot of children and their parents will appreciate being able to give their children the influenza vaccine differently other than a needle. So thank you for that. And thank you for sharing the difference of why maybe adults that wouldn't be as beneficial. We really appreciate that. Okay, so unfortunately, that is all the time we have. We could keep Dr. Kelvin here, I'm sure, all night with all our questions. So thank you so much, Dr. Kelvin, for not only your wonderful presentation and the way that you took so much information and complex information and made it in a way that we could understand. Really appreciate that. And for answering so many of our questions. We so appreciate you and your time and everything that you do each and every day. I also want to thank our sponsor, GSK, for making this webinar and our Lung Life webinar series possible. And to everyone that joined us, and thank you for your great questions and for all your comments. We're getting tons of feedback right now. Dr. Kelvin, so many people thanking you for your work and everything that you do, and appreciating your presentation. Many people are saying that they learned so much from you. So thank you so much. And everyone, I hope that you'll go out and get the vaccines that Dr. Kelvin spoke to you about tonight. And everyone stay safe, stay well, and breathe easy. Take care and have a wonderful night.
Thank you. Thank you to everyone too, and to LungsAsk.
Bye, everyone.